everybody. Hello, good evening. We're going to go ahead and get started. Hopefully you can see this okay. I'm just going to stand for a second. So thank you so much for coming to the October Big Valley Beekeepers Guild meeting. We're so excited to have you here in our monthly meeting. I know that you already let us know that you are new, and we are so glad that you're here. So please enjoy the evening. Make sure that you connect with each other because we have some great stuff planned, especially for 2023. We have a couple announcements before we kind of introduce our speaker. But thank you so much for supporting our host restaurant. This is our first meeting here, and we're really excited to be welcomed by Casa Flores, and hopefully this is a good upgrade for us. I think I'm seeing lots of nods, so that's fantastic. We are recording this meeting, but it will not be published on YouTube. It will only be available to members, and the reason for that is because our guest speaker is a researcher, amongst other things, and she may talk about some of her unpublished research. So we want to make sure that stays that for now, which is kind of cool. So let's see, just some housekeeping. There is a restroom back there. There's also another one through the restaurant and across in the back. And again, if you're interested in purchasing t-shirts or signing up, renewing, or joining, we can help you with that after the meeting. So, which is great. The good thing about Casa Flores is they're not closing at 8 o'clock, so we don't have to quickly rush. So just some news and updates. We are recruiting for a volunteer director in director role. So right now, our board of directors, we try to get to 5. But if you're interested in being a part of the Big Valley Beekeepers Guild and the direction and then all the exciting things we do that we're having to actually happen next year, please reach out to us. We'd be happy to share with you just kind of a snapshot, and you're welcome to join some of our leader meetings. So please, if you're interested, let us know. We also want to give a big shout out to all of our volunteers. And we've had volunteers thanks to Barbara, who is our volunteer coordinator. So if you'd like to help us with things like the spring class or any of the events that we put on, that kind of thing, that sort of thing, let us know. We'd love to get your helping hands. So speaking of booths, we actually have a booth opportunity on October 22nd in Stockton at the Ag Center. If that's something, we haven't been able to locate someone to be able to have a Big Valley Beekeepers booth there to kind of talk about what we do. But if that's something you're interested in and you're available that day, please email us and we'll get you hooked up with what you can take and talk about beekeeping. It's really easy and it's really fun. So you can tell it. Oh, we're selling something. Oh, okay. So if you're interested in selling something, I'll connect you with the person too. Gets that all taken care of. Okay. And then also we have a Christmas party. Our Christmas party is always in December. So we have one coming up on December 8th. If you'd like to help with the planning of that and the coordination, please let either Paul or myself know, or Barbara know. And we usually do a fun gift exchange. This is what we've done in some years, where everybody brings basically a wrapped gift, right, of some kind of beekeeping theme, and then we do a gift exchange. So hopefully you'll have that on your calendars coming up as well. So kind of cool. We actually were gifted these fantastic Big Valley Beekeepers Guild lapel pins. So if you join for 2023, we want to be able to give you one of those pins. If you're already a member, that's easy. We can help you with that. And then we'll have links probably in the coming weeks. If you haven't yet joined for 2022, then we'll help you do that for the remainder of the year and get you on board for 2023. And the pins are wonderful. In fact, I should be wearing one. Oh. All right. We also are so excited that we have t-shirts. So we've been wanting to have Big Valley Beekeepers Guild t-shirts for a long time. And they're super cool. We brought the whole box. So if you'd like those after the meeting, definitely get them with us. And we have all different sizes still available. So get them while you can. And then of course, if you'd like to sponsor a meeting, let us know. The Zoom fees are about $15 each month. So if you'd like to be a Zoom sponsor one of these days, let us know. And then, of course, we're thinking already about our Springfield Day. And we also participate in Ag Venture. How many of you have heard of Ag Venture? Yes. Oh, very good. So there's, there we go. So we always participate. We have a table set up at the one here in Lodi at the Great Festival Grounds. And then if we get enough volunteers to help, we'd also like to have a table set up for January in the one in Stockton. So let us know if you'd like to help with that. So with that, I'm going to sit down, but we're going to introduce our speaker. So we have with us Priya Basu, and she happens to be the assistant professor, pollinator health and apiculture, vice chair, early career professionals committee, Entomological Society of America, secretary and treasurer, and American Association of Professional Apiculture. Oh my goodness, I'm gonna have such a hard time with this, Priya. She's gonna laugh at me. So, oh my gosh, she is from Mississippi State University. So she's joining us late at night. So we're so happy. So we can all wave. So thank you. She has a lab website, and we'll be publishing this on the Facebook page along with the MSU website. But she's currently the assistant professor at the Department of Biochemistry, Molecular Biology, Entomology, and Plant Pathology. And she teaches honeybee biology and beekeeping. She's also a courtesy faculty at the Department of Horticulture, Oregon State University. Priya studies the interactive impacts of multiple stressors on bees and uses a wide variety or array of multidisciplinary techniques across fields such as physiology, toxicology, functional biology, and neuroethology to address her research questions. In addition to the research community, she also enjoys working with stakeholders, policymakers, and the general community in protecting bee pollinators and raising pollinator awareness. In fact, her desire for outreach and bee awareness has actually resulted in a children's book. And it's all on honeybees, so you can definitely take a look and maybe even purchase it. We are happy to say that the Big Valley Beekeepers Guild is going to make a small donation that we can do towards her research study in her lab. And what we'd like to do is actually challenge everyone here and those of you online to also go and make a donation towards her research in her lab. So if you'd like to match our donation, please let us know, or go ahead and do that. It's definitely worthwhile. She is, oh, sorry. Yes, I was just about to say it. Yes. So, and just so that you know, Priya is actually donating her time tonight for us and for the Big Valley Beekeepers Guild. And speaking of that, so thank you. So with that, I'm going to go ahead and stop sharing my screen. And Priya, if you want to go ahead.
Thank you, Sherry, and thank you everybody so much. This is really kind of you, and I'm so excited to be here. Virtually, I do have my water and sparkling water next to me, so I'm virtually joining you in the happy hour as well. I am going to go ahead and share my screen. Just let me know if you can see this.
Oh, yes, we can see it. We'll just expand it out. And just to let everyone know too, if you have questions, especially if you're online, go ahead and put them in the chat. And then we'll do a quick question and answer too. So at the end of her presentation. Okay.
Thank you, everyone. So today I'm going to talk all about exploring the many facets of honeybee nutrition. We know that there are multiple stressors that are affecting bee populations worldwide. We have pests, parasites, pathogens, diseases, pesticides, poor nutrition, climate change, and so many other different factors. Even though we keep talking about varroa, we talk about pesticides or other diseases, poor nutrition is an equally important stressor and still very much under-investigated. Nutrition is, of course, important because not just for us humans, for bees, for any organism, it is necessary to support life. For honeybees, specifically, we call nutrition to be the first line of defense. It is the key in dealing with major problems such as varroa, nosema, and other diseases. In fact, studies have shown that adequate nutrition helps improve bee health, bee longevity, bee immunity. This is just like us. If we are well nourished, if we have better immunity, we can also withstand stressors better. Same with the bees. And as I mentioned before, that nutritional stress, even though it is an important factor in colony losses, it is still not thoroughly investigated. There are many ways that honeybee colonies can be nutritionally challenged. There can be a lack of pollen abundance. Just the quantity of pollen that is coming in, that is not enough. There can also be a lack of pollen diversity. Too much of only one type of pollen is coming in. Bees do not have access to a diverse pollen spectrum in the landscape. There can also be a lack of pollen quality. For example, some of our studies have shown that blueberries, hybrid carrot seeds, their pollen is not as good of nutritional quality, for example, say as almonds or cherries or apples. And not just pollen, we also need to talk about the nectar flow. If there is an insufficient nectar flow, the colonies will, of course, be nutritionally challenged. But most importantly, when we are talking about bee colonies, overwintering is always at the back of our mind. As beekeepers, as bee researchers, we need our colonies to make through those winter months, which essentially means those hives need to have good nutritional resources stored in the colonies, as well as within the body tissues of the overwintering workers. We call them diutinous bees. So these diutinous bees, the longest living workers that overwinter, they need to be well nourished themselves so that they can survive the winter months and raise the new generation of brood the following spring. So all of these together really make it important that bees have access to a diverse flora in the landscape, both in terms of pollen as well as in terms of nectar. When we talk about bee nutrients in general, we can broadly categorize them into macronutrients and micronutrients. Macronutrients, as the name suggests, of course, it is very critical for bee nutrition and it is required in larger quantities. Examples include carbohydrates, proteins, and major lipids. Micronutrients, on the other hand, are equally important, even though they're required in smaller amounts. For example, we have phytochemicals, vitamins, minerals, phytosterols. So for every foraging trip, foragers will commit to any one of these resources and they will forage the landscape for this. So they could either be looking for pollen, they could be looking for nectar, or even water. And yes, water is also important. Not only because water is required to sometimes liquefy crystallized honey, but especially during the hot summer months, bees need water to stay hydrated. If there is a dearth of pollen in the landscape, bees will occasionally look for dirty water, what we call essentially is mineral-rich water. But water is also important. In terms of the proteins that bees get from pollen, just like the human diet, we eat a variety of different things to source different types of proteins, essentially the different types of amino acids that come in from this varied diet. Same with honeybees. They need a diversity in plants in their landscape. They need to have a wide variety of pollen types because each plant has different resources in its pollen as well as in its nectar. So the diverse the habitat is, the more different types of nutrients bees can bring in. And the same applies for proteins. We did, and we are constantly doing some of these studies. So we are analyzing protein contents of some pollens. I just wanted to show you some examples. Almonds, for example, is at 30%, meadowfoam at about 24%, hybrid carrot seeds at about 13%, rabbit brush 25%, corbicular pear, because we trapped it, so it had a mix of different types of pollen that we tested. So this was at 15%. Blueberries at about 27%, California poppy at 28%, sunflowers at 17%, and 2 different varieties of blueberry where we hand collected pollen from, we found that the Elliott variety has about 18%, whereas the Dukes have about 27%. Apples, 39%, cherries, 36%, apricots, 38%. So with that, it really tells us that different plant sources, their pollen, have a wide variety, a wide range of proteins that they're offering. Similarly, with the carbohydrates that bees get from nectar, they're mostly collecting sugar and they're storing it as honey. So bees can digest glucose, fructose, sucrose, trehalose, these types of sugars. What bees cannot digest are starch, corn syrup, dextrins. In fact, these 3 sugars, lactose, galactose, and raffinose, are poisonous for bees. If you are providing fructose, so if you are making your invert sugar, please watch out for this chemical, which forms as a byproduct called hydroxymethylfurfural, or HMF. So in nature, naturally, when bees are converting the sugars in the nectar into fructose, they do that using enzymes such as sucrase, invertase, and so on. HMF is not produced in this biological pathway. When we are adding something to the sugar syrup and we are doing it in a synthetic manner, this is when HMF is produced. And we have seen, in fact, there are quite a few studies about HMF as well, where bees start showing diarrhea-like symptoms. They have bloated guts and greasy guts. HMF in higher concentrations is definitely toxic for honeybees. And for a colony, say a worker bee needs about 11 milligram dry sugar per day. With a colony of 50,000 bees, that colony needs approximately 700 pounds of sugar per year. A flower, to the most, under best of conditions, would have 50% or less sugar concentration in its nectar. So you can imagine for that colony, how many flowers they really need to be able to source all of that sugar just by foraging. Lipids, another important macronutrient, it is still under investigated. Lipids in pollens can range anywhere between 1 to 20%. Examples, there are various types of lipids such as saturated fats, organic acids, terpenoids. However, what is really important is that you may have seen this picture before, where to your left is a forager bee and you're looking at the dissected abdomen. To your right is a nurse honeybee, same, a dissected abdomen. Look at the fat body comparison, which essentially tells you how much lipid stores they have. When your colony is ready to overwinter, your diutinous bees need to be somewhere close to nurse bees, even if not as well fat, as much as fat as the nurse bees, but the diutinous bees really need a lot of proteins and a lot of lipids within its tissues, within its body reserves to be able to overwinter. There are also a variety of micronutrients, as I mentioned, so many different types of salts and minerals that bees source from pollen primarily, like calcium, magnesium, zinc, and different chlorides. There are also vitamins A, B, C, D, E, and K that bees get from pollen. In addition, there are phytosterols that bees get from pollen, organic acids, phytochemicals, some phenolic acids that bees get from nectar. I do a lot of work on phytosterols specifically, other than looking at the different nutrients. This is because, you're in California, so you know that the almond pollination generates a huge amount of revenue. But to be able to pollinate that massive acreage, bees are forced to start their brood cycle earlier than they normally would. And beekeepers all across the country, they are definitely concerned about bee health and they appreciate the need for better nutrition. Studies have shown that colonies which do not have access to natural pollen and they're only sustaining on commercial crude protein diets, they do not perform as well as colonies that do have access to natural pollen. In fact, the major focus for decades has been macronutrients such as proteins and carbohydrates. Research on micronutrients is severely lacking. And this is where I come in. All insects are sterile auxotrophs, essentially meaning that they cannot synthesize their own sterols. So these sterols must be obtained from their diets. And you might ask me, why do you think sterols are important? It is important because sterols are components of cellular membranes, and they are precursors for important insect hormones, such as molting hormones. So without sterols and without steroidal hormones, insects are not going to molt, and they are not going to go into their different life cycle stages. All bees, honeybees, bumblebees, mason bees, all bees are sterol auxotrophs, just like any other insect. For bees, specifically, pollen is the only natural source of phytosterols. Even then, the type of phytosterol that is required vastly varies between the bee species. If we are only talking about honeybees, 24-methylene cholesterol is the phytosterol that honeybees require. Let's look at this animation video to sort of go over why phytosterols are important for honeybees. So pollen naturally has an abundance of sterols. When nurse honeybees are consuming pollen, they can selectively take up these sterols, and they are storing it within their body tissues as endogenous sterols. These endogenous sterols are then transferred to the growing larva through brood food. Thereby, the larvae have sterols, they can produce molting hormones, they can successively molt, and you have new generation of bees. If you're only feeding them these crude protein supplements which do not have these sterols, even though the nurse bees are eating it, after a point of time, there are no new sterols to replace the constantly used endogenous tissue sterols within the nurse bees. So after a point of time, no new sterols going into the brood, no molting hormones, and eventually you'll find a cessation of bees and new generations. There are no new generations. So this essentially tells us why sterols are really important. So with that, I have been doing over the past many years, understanding the impacts of phytosterols on bee physiology as well as overall colony health. So the research that I'm doing specifically looks at what are the different sterols, why 24-methylene cholesterol is important for honeybees, how much of it is required, how much to add in a diet so that that is the bare minimum your bees need to show improvement in overall colony health as well as physiology. So I have listed some publications here. And if you're interested, these are some of the papers. They are all open access, so you can read them. And if you have any questions, please do not hesitate to reach out to me. And everything that I'm going to just go over about bee nutrition in general, like macro and micronutrients, supplemental feeding, about the foraging behavior of honeybees in terms of nutrition, all of that was recently published in this open access journal. This chapter is called Honeybee Nutrition, and you can actually find all of the information that I'm talking about right here. So other than phytosterols, there are also phytochemicals that I mentioned, which are found in nectar primarily. For example, p-coumaric acid, camphorol, quercetin, and there are some studies that are coming out very recently which talk about how these phytochemicals can improve honeybee worker longevity. Of course, in the lab conditions, they can reduce nosema spore loads, they help counteract pesticide stress. So these phytochemicals, like other micronutrients, are very important, and it is really crucial that we understand how both macro and micronutrients have a role to play in improving honeybee physiology as well as their overall colony health. So if you're looking at your frame, if you look at the picture to your left, this is what we ideally aim for. Look at the larvae. They are floating in a luscious pool of brood food. These are typical of well-nourished larvae. To your right, the picture that you see, this is what a starved larva would look like. So the larva is, of course, in a very dry cell. There is hardly any brood food there, which essentially means that either the landscape doesn't have enough resources for them, and of course, which means that we need to intervene if we spot it early. So for fall hive management, we need to be able to prepare fat fall bees that are capable of overwintering for a period of between 5 to 6 months. The colony needs to have enough adequate reserves in terms of pollen and honey, as well as within the bee tissues, the nutrition that the bees have access to during fall will ensure that these bees are well nourished and well fed through winter. Bees that are emerging late summer or early fall, as I mentioned, must be well nourished. Protein must be stored in cells. And because the bees are clustering for warmth, we need to make sure that there is enough food resource within that cluster or close to that cluster. So depending on where you are located, your local weather, your climate, what your ultimate goals are, what's available in your landscape, how long can the bees have access to protein, to pollen, to nectar, the colony size, what are the types of bees, what are your aims in terms of what you want to do with the colony. So there are so many factors that come in. But if you would like to supplement protein during fall, you can either choose a pollen-based supplement or a non-pollen-based supplement. There are, benefits to both. So if suppose you just want your colony to have enough protein to last that fall and winter, you can potentially provide a non-pollen protein-based supplement. If you think that no, the weather is still warm and the colony can keep producing brood for a little longer, so you can potentially also feed them a pollen-based supplement, which will induce more brood production than a non-pollen protein supplement. So depending on what your ultimate goals are, where you're located, the local weather and all, you can choose the type of supplement protein if the colony needs it. Similarly, when you're providing them with sugar syrup, if you feel that your colony is light on honey, you can help build those hives. You need to provide 2:1 ratio, a thicker sugar syrup during fall, preferably use an in-hive feeder if it's too cold outside. Watch for first frost. So if you are in an area where you might get first frost, we need to then stop feeding liquid sugar syrup. And if additional sugar supplementation is required, then we can opt for granulated sugar, fondant, and other things. So during this transition phase from fall to winter, the bees are, of course, they're generating warmth within that cluster. They may continue to raise some young if they have some brood left over. They're, of course, using honey for food, and they're more concerned about clustering and protecting the queen. So the adequate population of healthy fat bees, the overwintering diutinous bees, is required to be able to survive that winter month. They need enough stored honey, some amount of ventilation so that there is no condensation and further cooling within the colony. And if there are warmer winters, you can also watch out for food resources in the hive. If you think it's warm enough to open the colony, inspect that they are not starving and you need to replenish any food reserves as needed, you could potentially try that. And if it's a warm winter, for example, where I am right now in Mississippi, in Oregon, it was an absolute no-no. I've never seen a warm winter, but here in Mississippi, there could be some really warm spells all of a sudden. Like last year, it snowed 3 days, but then there were super warm spells in between those 3 snow days. So we then resorted back to the fall formula of 2:1 sugar syrup. We, of course, need to be cognizant and leave ample honey in the hive for winter. If we take out more honey and if the colony feels a little light, we need to feed them immediately with sugar syrup to help them build that back up. And how much is enough in your region, that completely depends on where you are. So your local beekeeping groups, your local beekeepers, experienced beekeepers are your best resource. And in spring, if you remember, I showed you a couple of pictures about larval starvation. But in spring or late winter, if it's warm enough for you to open your colony, you see something similar to this. It is likely a sign of starvation in the colony. You'll have these bees with their head down in the cells, and they are dead in there because they were looking for that last drop of honey in those cells. Or you could find that entire cluster together, trying to locate food source, and they have all died. So these could be potential signs of starvation when you're ready to inspect your hive after winter. So essentially, what it means is that everything the bee needs is within its is found in this flower itself, in its pollen, in its nectar. We just need to make sure that the bees have adequate resources surrounding them in their landscape. If you remember this slide that I showed you, that how there are different factors which create nutritional challenges for bees, the new player in town is, of course, climate change. And being in California, you are no stranger to this. Climate change is increasingly contributing to uncertain weather patterns. We have higher temperatures, we have heat waves, we have drought, we might even have flooding. So this relationship that bees have with plants is very unique, and it's also very sensitive. Bees are dependent on plants for pollen and nectar, whereas many plants are reliant on bees for reproduction. With changing climate, this rapidly changing environment, there is a disruption of the plant pollinator networks, and it can happen in 4 ways. Either there could be a spatial shift where bees or plants are absent. So you have plants blooming, but there are no bees in that landscape. Or the other way around, there are bees in the landscape, but there is nothing in bloom. There can also be a phenological or a temporal shift. For example, flowers are blooming before bees emerge or vice versa. So in this scenario where ideally you would have seen bees, say, foraging from this undergrowth, there are no bees. So flowers have already bloomed before the bees emerged, or the bees have emerged before the flowers have bloomed. There can also be an adaptive, morphological, or anatomical modification of either the bee or the flower. Something has changed because of which that partnership is not working out. There can also be a change in the foraging behavior in the bee itself due to the climate effects. It is too hot for the bee to fly out and forage. It's too cold for the bee to fly out and forage. And some of our research, so we have already started working here, and I'm partnering with another scientist here at Mississippi State University. So we have started working on soybeans, and we are finding some similar changes with the extended heat and drought stress on the plant itself, as well as the nutritional quality of pollen and nectar. So this is just something that we have started, and I'm hoping by the end of next year, we'll have more data to show. And not just heat, drought, or flooding. There could also be other instances. For example, to your left, this is from one of our sites in Oregon, and what you see is a commercial blueberry orchard. So you already have colonies here, but this year it was a prolonged wet, cold spring. So either the blueberries are not being able to bloom, or even if they bloom, it's so cold, it's so wet, bees cannot forage. So this is clearly imposing nutritional stress on the colonies. And to your right, there, what it clearly shows that you could also have these sudden snow days. Everything stops, and the bees are, of course, they're unable to fly out and forage. And let's not forget wildfires. This is actually one of our ongoing experiments where we have sampled honeybees, workers, both nurses and foragers from colonies, as well as George here. He was very kind enough that even though he lost a significant number of his colonies due to the wildfire event, he gave us the surviving queens to test them out. Because what you see here is that there are these colonies that survived the direct path of the wildfire, but they were exposed to that extreme heat. And you can see him standing here where the burn patches are. So this is where some of his colonies once stood. So not just that, this is another video that was shared by another Oregonian beekeeper. So what you see here, no filter used, nothing. And of course, being in California, you are aware about the dangers and the effects of wildfire. So the bees are clearly very aggravated. They are bearding at the nest entrance. They cannot navigate. There is no sun, so they cannot forage. And when this goes on for a prolonged period of time, they are nutritionally stressed. And we had to intervene. We had to feed our research colonies, as well as we started helping our local beekeepers to help feed their colonies, because the bees were otherwise just stuck there. This was late summer, beginning of fall, and bees really needed to prepare for overwintering. To your right is that same area, very close to OSU campus. This was about 10 days or 12 days after the wildfire event, when we could actually start going out and checking the colonies. So this is something that we are currently doing, but just to throw this idea out that there are so many ways bees are nutritionally challenged, and now climate change is a new player in town. So the importance is to be able to provide a diverse forage for our bees. As I mentioned before, the diverse the forage, the more the types of macro and micronutrients that bees have access to. There should also be an abundant forage and a staggered bloom. So something or the other is constantly blooming throughout the year, and we need to be cognizant of which times of the year bees have a hard time finding pollen or nectar naturally in the landscape. So if we are able to provide a staggered bloom, something or the other is constantly blooming, this is really critical for the bees. And of course, as researchers, as beekeepers, when we take care of our colonies, we need to be ready to provide supplemental feeding when it is necessary seasonally. So with that, I'm just going to end by showing you three quick studies which are very related to nutrition. So as I mentioned, that we do a lot of work with poor nutrition. We also look at how different stressors interact with poor nutrition. So this is one such study. We are looking at how pesticides and poor nutrition together can impact honeybee colony health, individual bee physiology, as well as honeybee gut microbiome along an agricultural intensification gradient in Mississippi. And these are the two graduate students who are primarily heading this project. So what we're doing is east through west, we have selected 4 locations based on the use of pesticides, the agricultural intensification in these areas, as well as the availability of natural forage. And we have put out about 16 colonies in each of these locations. We are also creating these land use land scale maps, which essentially will also tell us that what is the natural landscape in these different field sites. This is just an example that I'm showing you from one of the 4 field sites, and each color represents a different type of habitat within that landscape. The star at the center is where our field location is, where our colonies are. And this circle has about a 3-mile radius. So we are looking at what that landscape within the bees' foraging range is. So for example, brown indicates cultivated crops, red indicates developed high-intensity habitats, blue indicates woody wetlands. So these are the light blue, of course, and the dark blue is open water. So these are some of the ways that we are characterizing that landscape. So what we did was, before we started the experiment, we need 64 colonies, but we started working with about 75 colonies because you never know what might go wrong. So we had to make sure that we equalized the colonies. They were all of equal strength, equal brood. Everything was similar before we took them out to the different field sites. We conduct monthly evaluations of every side of every frame in every colony. So we're looking at the adults, we're looking at the brood, we're looking at the queen. We're also sampling nurse honeybees from these colonies. And we are setting out pollen traps once a month. We are collecting pollen. We're also collecting uncapped nectar, uncapped honey from these colonies. So what we are going to do is, with the nurse bees that we have collected, we are counting mites in them. So essentially, we're trying to get an understanding of the varroa mites in these colonies. With these nurse bees that we have collected, we are also looking at their gut microbiome. We are doing various gene expression studies to understand their physiology. With the plants that are in that landscape, we are also trying to identify them. So we are trying to see what is in bloom in that area, at which month, what is happening that is close to the colony. So here you see something called a fusion strip, where we have this little jelly that we use to take a snapshot of the different types of pollen from the different surrounding blooming plants. And we're also slowly creating a database of these pictures that will be open access, available for everybody to see. And with the pollen and the nectar that we have collected, we are now conducting pesticide residue analysis to look at the actual field realistic exposures to pesticides in these colonies. We are also going to do nutritional analysis of the pollen, as well as use more of that pollen to ID the plant sources that the bees collected these pollen from. From the nurse bees, we're also looking at nosema, and we are going to repeat this field experiment next season with a few more other parameters. The second project that I wanted to share with you is creating the bee nutrition database. This was recently funded by USDA NIFA, and I'm working with Dr. Ramesh Sagili at OSU and 2 graduate students at OSU and MSU that we both share. So we know that habitat for bees, it is chosen based on the relative attractiveness of that habitat, but really not so much on the nutritional quality. And I keep giving my excuse. If you give me a bowl of French fries versus a bowl of salad, I will pick French fries over salad. Not necessarily because it's nutritionally optimal for me, but just because I'm attracted to it more. The same way, The habitat that we're choosing for bees, there are some wonderful recommendations. And of course, bees visit them, bees get the required nutrients from them. But do we really know what are those nutrients? Do we really know what's present in its pollen or its nectar? Even though we have some basic idea about the proteins, there is really not much information on the micronutrient profiles that are available for plants in North America. So it is important that we understand the nutritional profile of the commercially pollinated crops as well as the natural forage that bees have access to. We also need to understand the nutritional quality of the commercial diets that we provide to the bees, as well as the other non-crop plants that are available. So with that, we are now currently building this database to be able to provide feasible practical recommendations for both growers and beekeepers. And of course, the general community, policymakers, researchers. We are quantifying proteins and lipids from pollen. We are also using very specialized mass spectrometry-based methods to quantify amino acids, sterols, and other different micronutrients. We are hoping that this is going to be an open access database that will not only be in our individual lab websites, but we are also partnering with NRCS, USDA, to be able to add it to their plant materials database. So they already have a list of plants that they recommend based on your region, the time of the year, and which ones to plant. All we want to do is add additional information on the nutritional quality of the pollen from these plants. Right now, this is though a 3-year project, and we are aiming ambitiously to look at 100 plants. I really consider this as my lifelong project because 100 plants is nothing. And this database is geared towards all bees, not just honeybees, but also native bees, because once we are analyzing the nutritional quality of these plants, all bees will benefit. So just to give you an example, and this is also published in this journal called Metabolomics. So if you're looking at it, this is what I call as a pie chart. Each slice is a different color, indicating a different phytosterol. You don't have to read what phytosterols they are. This graph to your left is canola oil. This graph to your right is a commercial diet that we tested. If you just look at the colors, look at the slices, they are so different, which essentially tells you that each source has very different quantities and very different types of phytosterols. Similarly with amino acids, to your right is the corbicular almond pollen. To your left is corbicular almond pollen. To your right is the commercial diet. Same as before, each color is a different amino acid. If you're just looking at the slices between the two pie charts, look how different they are. Once more, telling you how different the amino acid profiles are between the two diets. So with that, we have partnered with Sam Droege at USGS Maryland, and we are using his help, his volunteers' help to hand collect more pollen. We have either improvised on existing hand collection methods, or we have made our new hand collection methods. But we are in the process of hand collecting many different types of pollens. We have also put this video together. I'm not going to run it completely, but essentially, if you're interested, I'll be happy to share this with you, where we are asking citizen scientists to capture honeybee pollen foragers, because we can rely on honeybee floral constancy, that they tend to visit the same type of flower in every foraging trip. So if you follow that bee and you're sure that bee is visiting at least 5 to 7 flowers of the target species, the plant that you want to collect from, and that honeybee has 2 large pollen pellets on its hind legs, we captured that bee using a sample jar. We briefly anesthetize it, dislodge the pollen, and then we collect it. So this way, we have over 54 citizen volunteers that have signed up to help collect pollen for us across US and Canada. And they have varied beekeeping experience. They could be master gardeners, they could be master beekeepers, they could also be just the general public. They have access to a variety of floral resources, but we are constantly running these virtual training sessions. We are training them on how to collect. We are providing some of the supplies. We're also providing them shipping labels to ship the pollens back to us. So if you are interested to help us build a database, please do not hesitate to reach out to me. So the take-home message is that Priya showed a lot of useless data and graphs. It could also be that we really need a variety of forage, because that is what I am constantly emphasizing: the diverse the forage, the diverse types of macro and micronutrients bees have access to. And if you would like to help us build a database, please reach out to me. With that, I'm also now trying to map the forage in the Southeast region, what I call as the Phenology Wheel Project. It is important that we understand what is in bloom in our area and when does it bloom. So I'm partnering with beekeepers in this local region. I'm asking them to trap once a month pollen for us for 48 hours. We will collect all of that pollen samples. We will not only a photograph to look at the color of that pollen. We will try and ID the plants. We will look at the nutritional quality, and we will keep taking notes. So if we are able to do this over the next 2 or 3 years, as long as we are collecting from that same region and the beekeeper commits to it, we can actually have some good data set to look at the nutritional profile of the different types of plants that are blooming in that area. We also have an understanding of the timeline of bloom. What is in bloom? Where is it in bloom? And what is the nutritional quality? This way, we can also track if there are any potential changes due to the climate effects in the plant phenology. So if you remember that this was one of the slides that I showed you, that there are so many different stressors that are working on bees. These stressors either act alone or they act in synergy with each other. And this is exactly the research that my lab does. So we are interested in basically understanding the nutritional physiology as well as the different nutrients and how they help shape bee health. So we are working on bee nutrition. We also work on bee toxicology. We want to understand the exposure to pesticides in the landscape, how it shapes. We are also working with climate change and how it's affecting bees. And most importantly, how all of these different stressors are interacting with each other. So we use a lot of different methods, both in the field as well as in the lab, to investigate each of these stressors individually, as well as to look at their synergistic impacts. These are some of the amazing graduate students in our lab who are really pushing hard. They're very enthusiastic. With them, we also have an army of wondergrads. I call them wondergrads because without undergraduate students, you really can't do much. And both the graduate students and I, we really rely on them. Some of them have already graduated in spring semester, but our lab is very new. So these students joined in January this year, and we are still expanding. With that, I would like to thank my long-term collaborators, all of our research and extension partners, stakeholders, funding agencies, without whose support none of this work would be possible. If you are interested in knowing more about our research and the other different types of things that we do, I have listed our Twitter handles as well as our lab website. I've also listed my email address if you're interested in reaching out to me later on. But thank you so much for inviting me to speak today, and thank you for your patience. I'm very happy to take any questions now.
Great. You know, that was a lot of graphs and charts, but that's okay.
I know, I was like, okay,
It was
late in
amazing.
the evening, it's a lot of graphs.
My brain is spinning right now, but it's, oh my goodness, fantastic information. So, okay, so we're gonna go through, we are gonna go through some of the questions in the chat first, and then Paul has the microphone, the other one. So I'm hoping this will work, because then we can get some more questions that way too. Yes. Right, I'll tell you after. Yeah. Okay. So there, so gosh, let's see. Okay. So first one is from Paul, and he says, will your research lead to better pollen substitutes? Do you envision that as?
I am hoping, because right now with phytosterols, we have found that 0.5% dry diet weight of 24-methylene cholesterol is really important for honeybees to have access to. So we are testing out different commercial diets, and we are looking at the phytosterols that are present there and how much of 24-methylene cholesterol is present. So I'm really hoping that this will help us eventually to make better pollen, or rather better protein diets. So I always say that, you know, if, I mean, of course, we need these commercial diets to feed our bees when there is a lack of pollen in the environment. But if you know, that is another reason why we are building this Phenology Wheel project. If you know, there is a time in the year when your bees are bringing back a lot of pollen, trap some. You don't need to starve your colony, but you can trap some pollen. Just keep that pollen in your freezer, mix it to the diet that you're giving to the bees. So this way, they are still getting a bit of natural, nutrients that come in from pollen.
Very.
Pollen will always have many different types of nutrients that may not be present in a commercial diet. So if you just even trap for a little pollen and you mix it up into your paddies when you give it to the bees, they still have access to that.
Got it. Good tip. Oh my gosh. All right, here's another question. Related to the previous question, how do you see or imagine that your research will be benefiting the beekeepers? What are all the different ways that it's going to benefit even us as backyard beekeepers?
That is an excellent question. Now, gosh, where do I start? Do I talk about my pesticide research? Do I talk about climate change and nutrition? I'll stick to nutrition for today. I'll stick to nutrition. So one, yes, we are hoping that we can help improve these protein supplements that we occasionally give to the bees when needed. So I'm hoping that we can help come up and formulate better ones. In terms of the bee nutrition database, we can actually provide some science-backed data, what you can plant in your area at what time of the year. So for example, if we have this database open to the public, including beekeepers, growers, everybody in the community, you're thinking, okay, I have, say, for example, Phacelia, borage, and bachelor buttons. This one has more proteins and this grows only this time of the year. How about I also add this plant to the mix, which can provide more lipids because this is higher in proteins, but this is higher in lipids. So then bees have access to both. This is just to give like a general idea. With the Phenology Wheel project, we are hoping to help make beekeepers make better choices, where to have holding yards, where to take their colonies, at which time of the year, what is blooming in their landscape. We'll also tell them if they need to plant something themselves or trap for pollen and provide it to the bees. We can nutritionally intervene during times of pollen and nectar dearth in an area. So that is also something else that it's at the back of my mind. But these are just like 3 random examples I gave. I can just go on talking, so I should be quiet now.
No, that was great. That was great. So gosh, so when it comes to winter bees, are winter bees produced as a reaction to shorter and colder days, or does it have more to do with the pollen availability?
Excellent question. Even though I'm inclined to saying that a bit of both, I would more talk about, it being shorter and cooler days, because the hive does start to slow down. The queen does start to slow down. She's laying fewer and fewer eggs, coupled with less available resources. I would say it's just years of evolution where both factors come in.
All right, very good. And then our last question in the chat is nosema common in Mississippi? Because you're in Mississippi, correct?
I am. Fortunately, not yet. So all of our field samples that we have tested, we started the field experiment from May. So we've started looking at the samples from May. I haven't found any nosema yet. I'm curious to see what happens in February and March. And this is my first field season here. I moved in August, but it was already towards the end of the season. So by the time we started doing any experiments, we set up the lab, plus the supply chain issue with COVID. So I struggled for the first few months to set up the lab. By the time we started everything, it was April and May. I haven't seen any nosema yet.
Oh, okay.
So I'm curious to see what happens with our February and March samples.
Yes.
But Oregon, yes, a ton of nosema during spring.
That was a big move to go from Oregon to Mississippi. It's a big, yes.
Yes. But you gotta go where the job is. I still absolutely enjoy working with Ramesh, and I'm fortunate that, I can keep going back to Corvallis. So I still keep traveling to the Pacific Northwest more and more. But yes, it's trying to keep a foot in both sides of the country.
Right. Very good. All right. So Paul behind me, wait, he's over. Wait, where am I pointing? This way. He's gonna walk around with the microphone and see if others have some questions. So yeah. All right. Anybody have any questions? I know. Come on, you guys.
Well, you have my email address, so you can all. Oh, I see a hand right behind.
Oh, there's a hand somewhere. Or Or,
You can always reach out to me with more questions later on.
So let me know if you can hear her. We're hoping this works. So, okay.
I can't hear her.
Oh, is the blue and green light on it? The blue light's on? Okay. Did you hold it really close? Keep talking. No? Oh my goodness.
I'm sorry.
Oh no, that's okay. Hold on. Yeah, take mine. Or yeah, or go to mine. Oh, there she is. Oh, there she is. Okay. Okay, we don't want these two close together though. There you go. Try that. Oh, the green light over there.
Can you hear me now?
Yes, I can.
Okay, so I was curious, how much of the depth of research have you guys done? Have you guys done research like across the country, like European countries or anything like in that sort, some sort?
Sorry, I missed the first part. I only heard European countries and that sort.
So how depth of the research have you guys done? Have you guys done anything like any database like across country, like in the Europe countries or anything like Australia or Hawaii or anything like that?
Excellent question. In terms of the database alone, we are just focusing on North America right now. Australia does have that, even though it's an older publication. Australia has Fat Bees, Skinny Bees by Somerville. So there is this free publication that is available. Quite a few years ago, this was done by Somerville. It's called Fat Bees, Skinny Bees. That does list some of the nutrients available in plant pollen. In terms of our database itself, it's just focused on North America right now at the moment. Even then, we are struggling with 100 plants. As I was saying, it is a lifelong project. But in terms of working with bees, my background, so I'm originally from India. So my PhD, part of it was in India, part of it was in England. So I've worked on Apis cerana, Apis dorsata, back in India, the two native honeybee species that are found only in the Indian subcontinent. I've also worked on bumblebees in England, and then I switched to Apis mellifera when I moved to Oregon.
Now, do each bees react differently for each pollen? Like that there's like certain types of different bees. So do each bees, I don't know if you've done any research like that, but has there been a research doing some sort of database where like there's, this type of bee reacts to this type of pollen versus this type of bee does not react to that much of a pollen or anything like that? Like the preference of whatever.
Excellent question. Excellent question. Yes, there are some studies which are coming out which are showing that depending on the bees, they may be looking for different things in the pollen. So they may be going for different types of pollen. And even in general, when you're looking at bees, they may be generalist bees like honeybees that can utilize different types of floral resources. There can be specialist bees, for example, squash bees, that only need a certain type of plants that they are reliant on. So even depending on that plant pollinator interaction, what types of pollen they are preferring, what types of plants they are going to can also vary. It's more about, what that bee is. Is it a generalist? Is it a monolectic? Does it only survive on one plant? Does it only survive on 3 different plants? So depending on their evolutionary context as well, a lot of these plant pollen preferences or plant nectar preferences can change between the bee species. This interaction can change.
That's all I have right now.
Oh, okay. Oh, no, that's great.
Thank you. Thank you for your questions.
Hold on, I'm going to try this one. All right, can you hear me? No.
Yes.
Okay. Oh, it did?
I don't know if I'm hearing you from this microphone or the other microphone. Okay, now I can. I can hear you.
How about now? You
Yes.
Can hear me now? Oh, there we go. Okay. We're having microphone difficulties. All right. How about anybody else with a question? Anyone over there? No? No? Okay. No? All right. All of our brains are spinning, so that's why. We'll digest and probably have more questions. So is it okay if we keep your we can post your email address and everything on the Facebook
Yes,
page?
absolutely.
Okay.
Absolutely.
We'll do. Okay.
Mississippi State is located where in Mississippi?
Oh, okay. Which part of Mississippi State are you located?
Northeast. I am about an hour's drive from Alabama. Tuscaloosa, Alabama. I'm about an hour's drive
Oh, okay. All right, got it.
from University of Alabama. I'm
Oh,
looking A town called Starkville, just like Corvallis, typical college town, survives on football and the university.
Oh, okay.
Just another college town.
That's awesome. All right. Well, oh my gosh, Priya, we appreciate, any last minute questions? Okay. Oh my gosh, we appreciate you spending your evening with us. We know it's late where you are, and
I appreciate you spending time with me, and thank you so much for the invitation.
thank you for being here for us. So we look forward to your research, and please keep in touch with us because we'd love to help support your lab, and there's some exciting stuff ahead. So thank you for all of the work that you do for bees.
Thank you so much. Thank you. You're very kind. Thank you so much. Have a wonderful evening, everyone. Thank you. Bye-bye.